you guys just need to kind of be familiar that the ribosome is pretty complex and what's happening at the ribosome. So the large part is where it has the 3 binding sites. So you'll see in the next slide or two, basically 3 transport RNAs are kind of working at the same time. Even though one's leaving, almost done, one's coming in, and one's really working. The small subunit is what binds to the actual messenger RNA, okay? The string of those A's, no T's, A's, U's, G's, and C's. I saw an analogy like a tape strip of the messenger RNA. Okay, now I think we can't label 2 yet, but let's see what can you label by looking at this. Okay, so number 1 is what? You guys with me?
Number 1.
What's number 1? Okay, so that one, and they're showing you the particular type on your diagram, methionine, because all proteins start with that one. So it's also the start codon. Okay, so anyway, we got that one. Don't have 2 yet. What's 3? The ribosome. What's 4? Okay, and you can see these 3 spots now. This transfer RNA has already dropped off its amino acid. It's exiting. It's attracted to its amino acid, though, so it'll go find its match, in the cytoplasm. And then this guy's doing its work. So what's happening, you guys, is when the transfer RNA comes in with the amino acid, Penelope, you guys, I'll talk to you after class. When it comes in, there's more of attraction between the amino acids. So the bond breaks here, and then that bonds, and you're getting your chain of amino acids. Okay, so let's fill in. Let's see, what can we get from this one? And now we have a little more complex here where you can actually see this code. But let me see, what are we filling in on this one, you guys? You could definitely get 2, correct? Peptide bond. We can get 7. Yeah, 7. We can get 6. Number 5 is the what? Start codon. Like I said, here's that methionine again. The code for it is AUG. Number 5 is the start codon. Number 8 is the anticodon.
I think that was in 2 slides ago, actually.
Okay. Okay, so this is what I want you guys to see, though. Here's your messenger RNA. Remember with the pop beads last class? So here we're reading the codon. You can use the genetic code table, right? That rectangle, first base, second base, and it'll tell you it codes for methanone. But there's actually this molecule that has that anticodon, and see, 2 and A goes a what? Hello? 2 and A goes a... Okay, let's go back to DNA. Everybody. A's bond to what in DNA? T's. In RNA, A's bond to U's. Do you guys see it? Is it way, way too small? A to U. U to A, G to, so see how it fits? Okay. See what's on the next one. Okay, so we add a little bit more here. So make sure you get anticodon down for number 8. I think we did that a while ago. We're doing the process boxes now. So number 1. Here's the second thing that's going to happen. The ribosome helps form a polypeptide bond between the amino acids, and it pulls the messenger RNA strand the length of 1 codon. Okay, so the last step is the now empty tRNA, because it's left behind, its amino acid exits the ribosome. The other one's making the bond. This one's coming in. Now, the interesting thing is, remember those stop codons? There's 3 codons that code for just stopping. That means they're not carrying an amino acid, and it's going to sever the building. It's going to be the end. So now that polypeptide chain, that chain of amino acids, is free to be moved to where it needs to go. It might need a sugar added to it. It might need some help getting folded up, maybe in the Golgi apparatus, maybe in the endoplasmic reticulum. And then the ribosome breaks apart, disassembled, that's what it says there, until it's needed again. Okay, so the last section for Chapter 8. And next week, no lecturing, and then we'll hit Chapter 9, and then after that you guys will have your exam. So mutations. Commonly, we really think of this as bad, right? Mutations are bad. It's not always the case. Normally it is the case because it's some sort of change. And it's like if you think about if you just dropped your toaster. The toaster wasn't working very well. Sometimes it worked, sometimes it wouldn't, and you accidentally dropped it. You pick it up, you need to make toast, you plug it back in, and hey, it's working. So maybe when you dropped it, 2 wires that were not always touching, maybe got slammed together, and now it works better. But most of the time when you drop your toaster, you're going to do damage to it, and you're going to make it not be able to work. Does that make sense? So odds are you're not going to make something better by dropping it. Does that make sense? So the same thing with mutations. However, you have to realize a mutation is just a change. Either the DNA was changed, or maybe when the messenger RNA was being transcribed, it messed up. So now there's a change in the code. Maybe when the transfer RNA is coming in, it really wasn't a match, but it got close enough there where it dropped off its amino acid. See, these are all changes, either in the code or in the protein structure. Most of the time, they're not going to work, but occasionally they'll work. Sometimes there's no change at all. Remember how the code is redundant? I'll just make it up. I could be wrong, but maybe like AAG might code for the same thing as AAC. You guys follow me? So the A is not supposed to be an A. It was supposed to be a C, but if it codes for the same thing, does it matter? You guys are spacing out. Okay, let me try that again. Listen to me. If it was an AAG, it got messed up, it was now an AAC. If it coded for the same amino acid, does it matter? No. Okay? So here a change happened and it had no effect and it didn't matter. So all mutations are changes in the code. Somewhere the code has been changed. So moving on here, we got a little bit of writing. I'm exaggerating. We got a lot of writing on this page, but not much talking though. So some mutations, the gene mutations, I'm sorry. Do you see the gene mutations? Is there a colon? Just write affect a single gene. So gene mutations affect a single gene. Now there's 2 bullets. I have 2 slides. So continuation with this one. The first example of a gene mutation is a point mutation. So a point mutation substitutes one nucleotide for another. You're getting that down in the bullet. So let's look at the example. So over here, and that dumb bar's up again, isn't it? So let's look at this is a mess up in the DNA. Now that's bad because that means the correct code can never be built of the RNA for this individual. So what is the problem? There's supposed to be TAA, and look at it's T, this has become a C. Am I in the right spot? Do you guys see that? So at one point, there's a different nucleotide there. It shouldn't have been there. The DNA polymerase, that enzyme didn't catch it. Now it's stuck. This person has this mutation in all their cells. Okay, but it's just one point, thankfully. It's only going to affect one gene. This is a gene mutation. Now, is there an effect? Because sometimes there's not, right? Because the redundancy in the code. So now what happened? So here we're supposed to have an A, we have a C, so we have a G here instead of a what? A U. And did it matter? Is this a different amino acid than this one? Yes. So this protein is going to have a different amino acid. It's probably not going to work. So that's a problem for this individual. Another type of gene mutation is worse. It's called a frame shift mutation. So second bullet, a frame mutation, right, you guys, inserts or deletes a nucleotide in the DNA sequence. So this reminds me of your scantrons. If you guys skip, right, you skip a number, you're supposed to bubble in 5, you missed it, and now you're bubbling 5 in on 6. If you keep going without noticing your mistake, they're all going to be off 1. You guys following me? Same thing, you were supposed to bubble in something on number 6, you bubbled 2 things on number 5, that's like a deletion. Now you're all off 1 again the other direction. So a frame shift. Now, is this more of a problem? Look up here. Is this more of a problem? Now it's not like it just messed up one amino acid, right? Now, so where's the mistake? In here, we deleted a G. See, there's supposed to be a double G here. So now it reads different. This is UCC, now it's UCG. Hey, this one's not so bad here though. Did it code for the same thing? Yes. But now we're still reading off every time. This is supposed to be AUU, it's GAU, we have a different one. And it's going to continue screwed up. Sometimes getting lucky, sometimes not. This protein's probably not going to work because it doesn't just have one mess up, it's going to have a bunch of them. You guys following me? Okay, so that's a frame shift mutation. Now, even worse than gene mutations are chromosomal mutations. So next to the colon, affect many genes. And then we have 2 bullets again, I think. Here's our first one. Gene duplication results from unequal crossing over. So what is this event when homologous chromosomes change pieces of themselves? What's that called? Crossing over. But if they're not just lined up perfectly, right, they could get unequal trading. And you can see that by the drawing here. So now this right here is duplicate genes. This guy is missing genes. So obviously that's going to be a problem, right? They're not going to be able to make the proteins they need. Now the second one is even worse, and that one's called translocation. These are just types of chromosome disorders. Translocation results from the exchange of DNA segments between non-homologous chromosomes. So now these guys, the purple and green, didn't switch. This one got confused and these guys switched. Well, they don't carry the same stuff. So when meiosis continues here, we're just going to be missing some of this green guy. Unless we got lucky and this one got also in the egg. But anyway, this is just a problem. The chromosomes are not correct. We could have cut a gene in half even when we did that. Now there's other types of chromosomal disorders that involve full chromosomes. Do you guys remember any of those on those karyotype pictures? Do you remember in the past? What's Down syndrome? Down syndrome is when you end up non-disjunction. The 2 chromosome pairs didn't split equally into their gametes. So now we got an egg that has 2 chromosomes when they're only supposed to have one of the pair. You guys following me? Okay, so sometimes, and then the other egg is missing a whole chromosome. So you guys need to remember that chromosome mutations affect lots of genes. They're going to have more effects. Can I go? You guys are doing great. We just got to do some copying and we'll be out of here. We'll be done with this soon. Okay, so here's your potential impacts. So just get all that down, potential impacts. Potential impacts, and I kind of talked about this earlier. Chromosomal mutations, right, they have big effects because they affect many genes. right? It's a whole chunk of DNA. Some gene mutations, though, you guys, so now we're getting down to just one gene, they can change the phenotype also. And here's some examples. So a mutation can cause a premature stop codon. So what if the change doesn't make it a different amino acid, it made it one of those 3 stop codons? Now you just severed your protein prematurely, right? You shortened your protein. It's not going to work the same way. So that's going to be a problem. A mutation may change protein shape or active site. So again, just one different amino acid might not let it fold properly. The active site, remember, is for enzymes, helping reactions happen. The last one, the mutation may change gene regulation. So there's other genes that don't code for proteins, but just tell genes to turn genes on or off. Okay? So check out, there's 2 examples. This poor gal right here has cystic fibrosis. And then she, for whatever reason, I don't know if there's an extra gene or what's going on here, but she makes too much mucus. Or maybe she doesn't have a gene that regulates that gene, or a gene that, breaks down, makes the protein that breaks down mucus. So it's difficult for her to breathe. And it's kind of like there's too much mucus in the lungs, like pneumonia when you get sick, and then the tissue's not working right, and you're not getting the oxygen. Now another one has to do with, you want to tell them about this one, Hager? What's the deal with this one?
Yeah. So it's an artery, and the blockage one is obviously like cholesterol and things building up, and it's restricting how much blood flow is going through, and that's why heart attacks happen. Well, in the no blockage one, the book was saying where this person makes a lot of the good cholesterol, which clears out the arteries and keeps them free flowing. So that could be, I think that's a good mutation to have, something that helps you from having heart attacks and things like that.
Cool. Okay, almost hanging in there here. Now, mutations, just in general, we're talking about you can have change, and then there's nothing negative happens because of the gene. So get these down, you guys. These are called silent mutations. Oops. Silent mutations. Okay, so silent mutations, just like the word implies, there's a change, but it has no effect. And again, I've been talking about them. One, the code is redundant, so maybe the same amino acid is plugged in. I haven't talked about this one. Lots of DNA is non-coding regions. So if there's a change there and it's not a gene, then there's no change. And actually, we can look at these because as organisms get more and more distantly related from each other, these silent mutations add up, and we can kind of use it as a clock to figure out when they branched apart. We'll talk more about that when we get to evolution. They may not affect the protein folding. So maybe it's a new amino acid, but maybe it's the same type of amino acid in the same family. Like maybe it's hydrophilic, it likes water. So maybe the protein still works. In body cells, they do not affect offspring. So we can get a mutation, right, from spending too much out in the sun, too much time out in the sun. Well, we're not going to pass that on to our offspring. It's just a mutation in our skin cells, not our eggs or our sperm. Natural selection often removes mutant alleles. So if the mutation is bad, where the person cannot survive, then they don't reproduce, they don't pass that bad gene on. Does that make sense, you guys? That's what survival of the fittest is all about. We're not talking about the strongest. We're talking about being able to survive and reproduce and pass on your genes. Mr. Hager was mentioning in last class that we're a little concerned here about the human population because we have people that are having babies that maybe weren't going to have babies in a natural situation. What else were you saying about that? You remember? Just the diseases. If we're curing diseases and the people are being able to live and have children, the children are still going to have the genetic disorder, so now they're going to have to be cured. So it's getting a little scary with our population. But some scientists would say, yeah, but we have this brain and we're able to do this. So that's also a form of being the most fit. Okay, so one more slide, I think, right? And you've survived. So this comes back to some stuff we've already talked about. You remember the vocab word mutagens? So it's just something that causes a mutation. So UV rays, chemicals can cause them. Smoking causes them. Some of the mutagens directly cause the genes to cause cancer. So then they're called carcinogens. Do you remember that? So, and then here's a positive sort of thing here. We can use mutagens, which hurt DNA, which hurt cells, to fight cancer, because cancer is our own cells that are messed up. So some cancer drugs use mutagen properties to kill cancer cells, like radiation, chemotherapy. All right, so you guys hopefully survived Chapter 8. Oh, this is 8.4. This is transcription. Now a little bit of review about last lecture. Last lecture I taught you about a different process. The process of DNA. DNA synthesis is one way of saying it. It's not the actual name of the copying process. That's what I'm looking for. No, maybe page back and look at your power notes last class. So when DNA copies itself, what do we call that? Everybody turn the page back. It says it. What's it called? What's the name of the power notes? Replication. So do you guys remember? I taught you how DNA copies itself. What is needed? What is the situation between these two molecules that allows it to copy itself? A's always bond to T's. C's always bond to G's. So what would we call that? Pair bonding, right? There's pair bonding rules. Okay, you guys remembering this at all? Okay, look at your keywords over here. So replication was last time. We'll talk a little bit about it. We'll do some modeling later. But then we're going to go on to transcription and translation today. Then we'll do some more interesting stuff, and we'll make some models. And then we'll play a game. Okay, so transcription converts a gene into a single-stranded RNA molecule. So we've been talking a lot about this. What's this again? DNA. Today we're going to talk a lot about RNA. That's the other nucleic acid that you need to know about. Well, it's the only one I know about. RNA and DNA, that's it. Okay, so let's look at this flow chart. So right away, the central dogma. I don't really care that you remember that term, but this is the biggest thing. This is how molecules turn into life. So number one you see is DNA. So you can do your products first, or you can kind of go in order. Yeah, do your products first. So DNA arrows to what? RNA. So that's number 3. So then we got RNA converts and turns something, brings up some proteins. Now, those are the products. Here are your processes. So DNA copying itself, you see this loop? That's called replication. RNA being copied from DNA is called replication. And then the RNA turning into or coding for the protein is called translation. So we should have our flow chart completed now. Now, I wrote this over here. Let's just put it on the side of your notes somewhere. Proteins are chains of amino acids. The chains of amino acids are known as polypeptides. It's really wordy. This whole section is wordy because it's really complex. You guys need to remember way back when I taught you about the 4 big macromolecule groups, like carbohydrates. Do you guys remember what carbohydrates are? Close. Carbohydrates. sugars. Lipids, right? Fats and oils. Then we have proteins. And then we have our nucleic acids. Those are your 4 macromolecules of life. That's what life is built on. Now, these were the nucleic acids. Do you remember last time I told you that the monomers, the building blocks of DNA, of these nucleic acids, are called nucleotides. Okay, Michelle, put that away and listen to me. These are called nucleotides. They build the nucleic acid, they build the DNA. Proteins building blocks are what? Amino acids. There are 20 different amino acids. And they are put together in a chain. And we call the chain a polypeptide. Now, I'm not going to make you memorize that word, but when you read it, hopefully at least that you're kind of recognizing it. So if I was to draw a little thing of polypeptides, I would be linking together amino acids. So there's a triangle shape amino acid. It's bonded to a circle amino acid. These are just symbols of different amino acids. Oh, here's another one of those again. Now, why do they call it a polypeptide? These are called peptide bonds. And there's a bunch of them. Poly means many. Okay? Again, just want to introduce you to these vocab terms. Even if you don't really remember the exact definition of them, at least they won't scare you when you're reading, and you can read around them to solve problems. Okay, back to this again, this overall thing. The DNA is stored in the what? Nucleus. So the big thing about the RNA, and this is specifically going to be messenger RNA, is it's a messenger. The code is stuck in the nucleus. So we make a copy of it in the form of this molecule. This thing can move. It goes out into the cytoplasm where it needs to go to the organelle that makes proteins. Does anybody remember the name of the little organelle that makes proteins? These little dots. Ribosomes. Ribosomes. So now the messenger RNA is going to head to the ribosome where the next process can occur, which is translation. The ribosomes need the instructions. They need the code. So RNA is the link between DNA and proteins. Okay, let's fill in this next table. Okay, so DNA, last class, besides doing replication, the first thing we went over was the structure of DNA. So the first box, you should know that the sugar in DNA is called what? Deoxyribose. That's what the D stands for. Now you knew it had thymine. What were the other 3 nucleotides it has? So it has thymine, give me the letters, A and CG. And this bonds to that and that bonds to that, correct? Okay, but check this out. RNA has uracil. It doesn't have thymine. Something kind of different, so that's why it's saying that, okay? Also, you should know that DNA is double-stranded. You should remember the name of it. It's a double what? A double helix, okay? A spiral. Now, okay, what's the deal with RNA? The R stands for what? Ribose. That's the name of its sugar. So it's ribonucleic acid, okay? It does not have thymine as its 4th base. It has uracil instead. And it's only single-stranded. It's also temporary. It's built in red, and then recycled. Okay? It can be used over and over, too. Okay, so now, when I have these instructions, you guys, of labeling 4 through 7, it's going to take 2 pictures. So just label what you can from this picture. So this big blob is got RNA polymerase in it and a couple other enzymes. I don't need you to really worry about it, but these are the enzymes that are allowing transcription to take place. They're building the messenger RNA. Now, the start site, I think this is more interesting. Genes, where are genes located? Hello? Genes are located on the DNA. They're a sequence of the DNA. They're a piece of the DNA, you guys, right? Okay, so all of a sudden the cell's like, hey, I need some more hemoglobin, right? I need some more proteins to collect the oxygen in my red blood cells. So then the enzymes go find the spot that codes for making hemoglobin. And that's the start site. So it connects to it. It's an enzyme. And it unzips those hydrogen bonds. It's opening up the code for the DNA. Not for it to copy itself like last time, just to reveal the set of instructions it's looking for. So here it starts. It attaches. It unzips. It breaks the hydrogen bonds. There's nucleotides waiting. A's, C's, G's, and U's this time we need. Okay? So then let's move to this part. Okay, so actually let me back it up for a second. What? Why are you whining? Okay, I'll come right back. I just want it to flow nice. So, join me, people. The the transcription complex, the enzymes, once again, are binding here, breaking the hydrogen bonds, getting it ready. Number 2, the polymerase, the RNA polymerase, the specific part of this enzyme, this is a big complex, reads only one side of the DNA. Okay, it's not like with DNA replication. The code only goes in one direction, just like our language. We can only read our words one way. And then it builds the messenger RNA. So if this is a T, what is going to go there? Everybody join me. Stop writing. Listen to me. If this is a T, they're going to put an A there, right? If this is a C, they're going to put a... So just like with replication, they're building the complementary side, but this is a single strand. It's RNA. Wherever a T would go, a U will go. Okay, so it reads one side. We've got this stringing together the messenger RNA. 3, the growing messenger RNA hangs freely and detaches once the gene has been transcribed. The DNA helix winds again. Okay, now before I go on, do you have the messenger RNA labeled? Do you have the DNA labeled? We're okay? Okay, so then we continue, and there's no words for it, but then it disconnects, right? It was basically just using the template to be made, and it's mobile. This is messenger RNA. This is showing the complex breaking up into its individual parts. It can be reassembled, parts can be used so that it can happen again. Okay, there are 3 types of RNA. So please get that down.